Okay, so today we will be discussing amino acids, okay, or to be very specific, we will be talking about amino acid chemistry. Now, before we proceed, I want to ask you a basic question. How many amino acids or standard amino acids are there in this world, in this whole world? Okay, now, yes, how many amino acids are there in this world? Standard amino acid, yes. Okay, 22. Here 20, okay, 22, 20, 20,000, okay, 20, okay. Now listen, I am writing in the board how many amino acids are there in this world, you know. Unbelievable, right? Right? Unbelievable that I am saying there are 900 amino acids are there in this world. Why I am saying this thing? Have I gone crazy? No, absolutely not. Why? When I was asking you the question that how many amino acids are there in this world, you were unaware, totally unaware of the fact that I was asking actually how many standard amino acids are there. So probably you don't know the definition of a standard amino acid. Okay, listen. Amino acids we will talk about in details today, but first we should know what do you mean by amino acid. Listen, there are basically two types of amino acid, primitive classification. Okay, there is standard amino acid and non-standard amino acid. Non-standard amino acid. Okay. From now onwards, we will write amino acids as AA. Okay. What do you mean by standard amino acids? What do you mean by standard amino acids? Any amino acid which is incorporated into a protein by the help of genetic coding is called a standard amino acid. Okay, is called a standard amino acid. That means how many are there according to the definition, that there are actually 20 standard amino acid. Okay, now listen, but when I am talking about total amino acid present in this world, then it will be around 900. Okay, why? Because what do you mean by standard amino acids? You know, those amino acids are called standard amino acids which will be, you know, incorporated in a protein by the help of genetic code. That means they are produced, or they are, you know, helpful for the protein production. But out of this 900, only 20 are useful, right? 20 plus 2 are useful. Other are not at all useful for producing proteins, right? Now listen, you should first understand, that means you have told correct thing, right? You have told actually correct thing, that is there are 20 standard amino acids, but without knowing what are standard amino acid. To know exactly what are standard amino acid, you should know what is non-standard amino acid, you know. Listen, what do you mean by non-standard amino acid? What do you mean by non-standard amino acid? Non-standard amino acids are those which are coded by stop codons, which are coded by stop codons. Okay, which are coded by stop codons. So, which are coded by stop codons. Okay, now listen, what are the examples of non-standard amino acids? There are two main examples of non-standard amino acids. Number one is selenocysteine. Number two is pyrrolysine. Okay, number one is selenocysteine, number two is pyrrolysine. So these two amino acids are coded by stop codons. Okay, now you should always understand that we will actually discuss amino acid, or we will actually divide our amino acid lecture into three main segments. Okay, number one is we will at first discuss about the structure of amino acids. Second, we will discuss about the classification of amino acid. Third, we will talk about titration of amino acids. Okay, step by step we will go. Okay, structure of amino acids, classification of amino acids, titration of amino acids. Okay, so this is the right time to learn the exact structure of amino acids. Now listen, you should always understand that whenever I am talking about amino acid, first of all you should understand that there are actually two groups. Okay, listen. Listen, when I am talking about amino acid, you should always understand that, have you ever read organic chemistry? You have read, obviously you have read in your 12th standard or something like that, right? Now listen, what do you mean by this term, amino acid? Listen, from this term we know that this is an organic compound. Having two groups in it. Number one is NH2 group. Number two is COOH group. Right? Number one is NH2 group. Number two is COOH group. Now listen. Why it is called amino acid? Now you again must be thinking that I am crazy, that I am asking some stupid questions, right? That is, what is the meaning of this term, that why it is called amino acid, right? But probably you don't know this. Listen, why it is called amino acid? Why not acidic amine? The question is this: why it is amino acid? Why not acidic amine? Because if you have read your organic chemistry books, you will understand that it is called acidic. It is not called acidic amine. It is called amino acid because here the acidic group is the primary functional group. And here amine is the secondary functional group. Okay, that means, you know, there was a list in your organic chemistry books at first that during nomenclature, who will get the first preference? Carboxylic acid, then aldehyde ketones, then comes alcohol, then after that amines, right? That's why these are called amino acid. Why? Because it's a IUPAC nomenclature rule that you will have to take the name of secondary functional group first. You will have to take the name of your secondary functional group first, then you will have to take the primary functional group name. Okay, this is the basic rule. Okay, you should always understand that during IUPAC nomenclature you will have to take the secondary functional group name first, then you will have to take the primary functional group name. Okay, am I clear to everyone? Okay, let's proceed. So that's why it is called amino acid. Okay, now what is the basic structure of an amino acid? Listen, very easy way to draw any amino acid is, you need to understand that a very easy way to understand or draw any amino acid structure is that you will have to draw a carbon, okay, and you will have to draw its 4 valences. You know, carbon has 4 hands, right? Now listen, you need to understand that you will have to draw a carbon with its 4 valences, and in one of the hand you will have to add the NH2 group. In other hand, you will have to add the COOH group or carboxylic acid group. Then in another hand, you will have to add one H, and in another hand, you don't have to add anything else. You will just have to write R. You will just have to write R. Now listen with all your attention. Suppose if I say that what is glycine? If I say what is glycine, and if I say what is alanine, so what you will say is these are both amino acids having different R groups. That means amino acids are named according to the different R groups they have. Amino acids are named according to the different R groups they have, right? It's true. Okay. Now listen with all your attention. That means R group plays an important role in nomenclature of amino acids. It's clear now. Now listen with all your attention. I want to test your knowledge now. You people are very much knowledgeable. Okay. Oh, you are laughing. Thank God. You understood that you are not. Okay. Now listen. Suppose if I tell you that what is the function of this R group? What is the function of this R group? If I tell you that what is the function of this R group, what you will say? What is the function of this R group or side chains? What you will say? It helps in titration. Okay, this is a very new concept for me. Thank you. What is the use? It helps in pH balance. Partly right, partly wrong. Yes. Helps to maintain the structure from where you have read it. It's true. Okay, now listen. Many answers are coming, but listen with all your attention. What is the real truth? R group has varied number of functions. First function is this. Suppose one amino acid called histidine. Histidine has a R group. Okay. And this R group has two main functions, What do you know? Listen. Histidine are present in many enzymes active center. Active site? In an enzyme there is one active site, okay? Sorry. Where substrate binds, right? In this active site it is seen that many enzymes have histidine. Okay? So it is said, and now it is known also, that the R group of histidine actually helps in catalysis. That means it undergoes reduction or oxidation. And the second thing the R group of histidine does is it is actually helping hemoglobin to release or to take oxygen. Okay, that means it helps in what? pH control. It helps in pH control. It helps in pH control as well as it helps in catalysis. So there are many varied many varied functions of this R group. I have just given one example. Okay. Now listen with all your attention. Okay. Suppose this is an amino acid. Okay. Now, if I ask you what is the average weight of an amino acid? Yes. What is the average molecular weight of an amino acid? You don't know and you are laughing. Great. You are so much confident and ignorant about your knowledge. Very good. Ignorance is bliss sometimes. Very good. Now, what is the average molecular weight of an amino acid? Now listen. How to find out the average molecular weight? How to find out the average molecular weight? If I tell you, find out the average molecular weight of an amino acid, what you will do? You will just tell me, give us the molecular weight of every amino acid from 1 to 20, right? Right? Then what you will do? Suppose this is M1 and it is M20th. Okay. Now what you will do? You will just what? Do an average M1 plus M2 plus M3 or M20 divided by 20, right? This is what you will do. Yes. This is what exactly every biochemistry student and every biochemistry professor actually did many years back. And they found out that if we do that, if we just take the molecular weight of every amino acid and divide it by 20, then the average molecular weight would have been is actually 136.9 Dalton. Okay, 136.9. But now I am giving you a surprise. Why? Listen. In our day-to-day calculations, in any laboratory, in our day-to-day calculations, we are using only 128 Dalton as the average molecular weight. We are using 128 Dalton as the average molecular weight. Why? Can anyone say why? Why? Why do you think? Listen, this is the actual, exact actual, listen, this is the actual molecular weight of an amino acid. Now, why we are using this for our calculation? Why we are not using this? Just because it is a fraction? It is a fraction? No. Why? Why do you think we are using it? Listen, this is actually a standardization. Okay, standardized methods. This is the standardization of this process. Listen, you should understand that when biochemistry professors came up with this number, then many argued that many biochemists argued that most of the amino acids are low molecular weight. Most of the amino acids are low molecular weight. Most of the amino acids are low molecular weight. That means this number is actually false. Why? Because suppose M1, M2, M3, M4, M5, M6 up to 11, suppose 11 amino acid, the molecular weight of 11 amino acids is not much more, okay, but the rest of the 9 or rest of the 5 or 6 amino acids molecular weight are very much high. That means the average is increasing because of those only 5 amino acids, no? That's why biochemists have come up with this number, with this standardized number, that after many calculations, that 120 Dalton is actually the average molecular weight of any amino acid. Okay, right? Now there is a surprise for you all. Okay, wait for the surprise. Listen. Now listen. For any amino acid, listen, the average molecular weight is 128 Dalton, right? Now there is a surprise for you all. Listen. Suppose this is, don't worry, we will have a separate class on protein chemistry. Okay, for the time being you just understand the basic concepts. Okay, so understand this. Listen. So listen, we have produced, by interaction of two amino acids, we have produced one peptide. Now listen, dipeptide. Now listen, this is what? This is an peptide bond, right? This is one peptide bond. This is one peptide bond, right? This is one peptide bond. Now listen with all your attention possible. Whenever you are actually sticking 2 amino acids or 3 amino acids together, that means whenever you are producing 1 or 2 peptide bonds, you should always understand this. Whenever you are sticking 2 amino acids together, that is when you are producing a peptide bond or 2 peptide bonds. In each case, one water molecule will be going out. One water molecule will be thrown out, right? Now listen, why I am saying this, because whenever we are talking about these things, peptide bond, you should always understand that, suppose if I tell you what is the molecular weight of this dipeptide, suppose if I ask you what is the molecular weight of this dipeptide, okay, now what will be your answer? Your probable answer will be 128 into 2, right? That is what? 256 Dalton, right? It's true if you are an idiot, right? Now listen, to really find out the molecular weight of this peptide, you will have to subtract the molecular weight of water, that is 18 Dalton from it. Okay, that means what is the what is coming out? 8, right? Oh my god, I am doing maths, good. 8, 1, right? 2, 3, 238, am I right? 238 is the answer. That means, listen, every time you consider 128 Dalton as the average molecular weight of an amino acid, you will have to subtract 18 Dalton from it if it is participating in peptide bonds. Now listen, during exam it's very much complicated for you to understand that how many peptide bonds are there, how many, it's very complicated. Okay, suppose if I tell you that in a pentapeptide, suppose what is the molecular weight of a pentapeptide? What is the molecular weight of a pentapeptide? What will be your answer? 128 into 5 minus 18 into 5. So you are an idiot if you are thinking that this will work in your exams, right? Listen, this won't work. During exam, when you are asked that what is the molecular weight of a pentapeptide, the prompt answer will be 110 into 5. That's it. Simplest thing. 110 into 5 plus 18. Why I am adding this 18? Can anyone say? Suppose if during exam you are asked that what is the weight of a pentapeptide? Suppose it can be hexapeptide, it can be anything. You are intelligent enough to understand. So what is the weight of a molecular weight? Molecular weight of a pentapeptide. So it will be 110 into 5. Okay, that means what? 550 plus 18. That means what? 568. Right? Now tell me why I am adding these two. Why I am adding these two, these 18. Listen. Why I am adding these two? Suppose if I ask you what is the weight of a pentapeptide, then your prompt answer will be 110 into 5 plus 18 Dalton. Okay. Now this will be 568 Dalton. Now my question is why I am adding this 18? Why I am adding this 18? Because you should understand the basic thing that suppose this is a peptide, Okay, suppose this is a dipeptide. Okay, suppose this is a dipeptide. Suppose this is a dipeptide. Okay, no, this is no. Listen. So you are confused. Don't worry. I will show you. Don't worry. Suppose this is a dipeptide. Okay. Okay. He is very much, good student of mine. That's why he's saying that draw the pentapeptide. Okay. I won't be drawing the pentapeptide, but I can try tripeptides. Why? Because pentapeptide will be so much big. Okay. Now listen. Okay. Okay. So this is your tripeptide. Okay, listen. This is your tripeptide. Okay. This is your tripeptide. Okay. Now listen. If this is your tripeptide, then how many peptide bonds are there? How many peptide bonds are there? Listen. How many peptide bonds are there? There are 2 peptide bonds. There are 2 peptide bonds. That means when I was talking about, listen with all your attention possible. When I was talking about pentapeptide, there were actually 4 peptide bonds. 4. Okay. That means how to find out number of peptide bonds? How to find out number of peptide bonds? Peptide length, that means whether it is tripeptide, dipeptide or tetrapeptide, whatever it is. Peptide length minus 1. That's it. So this is a tripeptide, that means peptide length is 3 minus 1. Okay. 2. There are 2 peptide bonds. So these are your sweet and sexy peptide bonds. Okay, now listen. So these are your peptide bonds. Now listen with all your attention. Now what we will have to do is, we will have to understand why I was adding 18, right? Listen, this is actually your tripeptide, and these H and OH are associated with this carbon and this N, right? Now listen, why I was adding 18? 18 is the molecular weight of water, right? Why I was adding 18, because during when I was using the term tripeptide, dipeptide, in a peptide molecule, the terminal N and terminal COOH exist in non-ionic forms. That means complete OH and complete H are there, which are absent in what? Peptide bond formation. Because during peptide bond, this H and OH has gone out, right? But in a complete peptide, this OH and H groups are present intact. That's why we will have to add their molecular weight as well. Okay, we will have to add their molecular weight as well. Clear? Okay, so this is the basic of peptide length calculation, and we will, don't worry, we will actually get into details of these things when we will talking about proteins. I was just teaching you because I was actually into amino acid molecular weight, because you should know that what is the average molecular weight of an amino acid, 136.9, but what is the calculatable average molecular weight of an amino acid, 136.9. What is the basic average molecular weight of a amino acid which we will use for working in a laboratory, 128. But in an exam you will have to know 110 into number of peptide bonds, that means 110 into peptide length, that is tripeptide, 110. So what is the molecular weight of this tripeptide? 110 into 3 plus 18. What is the molecular weight of a pentapeptide? 110 into 5 plus 18. Why I am using this 18? Why I am adding this 18? You have already discussed. Sorry, we have already discussed that. Okay. So up to this it is clear, right? Don't worry, we'll go into the details of these things. Okay. We'll go into the details of PI and all these things, PI of every protein. Okay, possible. When we will talking about proteins. Right now we are discussing about amino acids. Okay. So we have already discussed the average molecular weight of an amino acid, that is 128 kilodalton, sorry 128 dalton or 110 dalton. Now listen, now we will be discussing some conformations and configurations or stereoisomerism of these amino acids. Okay. Now listen, suppose this is an amino acid. Okay. Suppose this is an amino acid. Okay. Now, can you tell me whether it is D or L isomer? Now listen. If you don't know D and L isomer, I think you should go back to the video where I have taught carbohydrates. You were absent that day. That's why you are lost today. Very good. Listen. So what do you mean by D and L? D means dextrorotatory? No. D is a form of isomerism which shows in amino acid D stands for dextro, right? Wrong. In amino acid D stands for where the NH2 lies. Now listen, if the NH2 is on the right hand side, then it is D amino acid. If in this amino acid the NH2 is on the right hand side, then it is D. Now how to draw the L amino acid? Okay, no need to draw these things. Now listen, this is L amino acid. Okay, this is L amino acid. Now problem is this, you need to understand, that is, how will you know that this is L amino acid or D amino acid? Listen, in an L amino acid, the NH2 group is on the left hand side. In a D amino acid, the NH2 group is on the right hand side. Now very important point you need to understand here is this, that cell specifically produces L amino acid and D carbohydrates. Okay, a living cell specifically produces L amino acid and D carbohydrate. Okay. Now, cell, okay, let us state it like this, that cells predominantly, that means most of the time, predominantly produces L amino acids. That means cell specifically produces this type of amino acids more than this. Now listen, but in biological system you will find some D amino acids. You will find some D amino acids in nature. Where you will find it? My question is, where you will find D amino acid in nature? In nature, where you will find D amino acids? There are some marine snakes. Okay, what is a snake venom? First tell me, what is a snake venom? A snake venom is nothing but a cocktail of enzymes, hydrolytic enzymes, which are lysines in nature. That means they actually cleave fibrinogen. That's it. That's why your blood clots. That's it. It's nothing but an activator of clotting factor and fibrinogen. That's why your blood clots and you die because of heart attacks. Now listen. In marine snake venom, there is one thing that which is known by the name of country fawn. Okay. In marine snake venom, there is a there is a component by the name of country fawn. Country fawn. Okay. So there are some marine snakes. Okay. Their venom has D amino acids. Okay. There are some marine snakes which have D amino acids. Okay. Another thing. You know gram positive bacteria? Gram positive bacteria also have D amino acids. Okay, also have D amino acids. Now listen. You know something called peptide? You know something called peptide? What is peptide? A peptide is an arrangement of a peptide where L and D amino acids are present alternately. You know NAG, you know N-acetylmuramic acid, N-acetylglucosamine. In bacteria, N-acetylglucosamine, N-acetylmuramic acid. Suppose this is N-acetylglucosamine, and then it is N-acetylmuramic acid. Then after that, N-acetylmuramic acid. Alternately, NAM and NAG are present, right? Now listen. In this structure, suppose this is your bacteria. This is your bacteria, and here it is NAM. Suppose NAM NAG. They are connected. Now listen. In bacteria, these NAG, things are connected via a tetrapeptide, and this tetrapeptide is actually a peptide. That means this tetrapeptide is actually made up of L amino acid, then D, then L, then D. Okay, clear? L amino acid, then D amino acid, then L amino acid, then D amino acid. Okay. Now listen with all your attention. Whenever I am talking about this peptide, you should always understand that these peptide, in peptide, the D amino acids are present. That is unique. Okay, and that's why if any cell disrupts this membrane, these D amino acids comes out and they will become potential antigen. Okay, clear? Okay. So now we have discussed the conformations and configurations. Now understand this very basic thing, that now you know what is D and L amino acid. Now you should always understand that this D and L amino acid has no connection with positive or negative or small d and small l amino acid, that is dextrorotatory and levorotatory. Okay, I have already discussed in carbohydrate class that what are dextrorotatory, what are levorotatory sugars. If you don't understand what is dextrorotatory, levorotatory concept, go and watch that video. Okay, now listen. So now we will be entering the classification of enzyme part. Okay, now listen. So before entering the classification of enzyme part, you should understand that if you go to any website which deals with protein structure or something like that, you will, if you just see, if you want to see the primary structure of any amino acid, or sorry, primary structure of any protein, you will see like this. Okay, you will see like this: P, S, T, L, M. That's it. you will get this. Not only this, you will get long chains, T, M, L, L, Q, and it goes on and on. What are these? These are called, this is actually a protein, okay? This is actually a protein, okay? Now listen. Now why I am saying this is a protein? Because it's a 1, 2, 3, 4, 5, it's a pentapeptide. What? Yes. The single letter code actually is a symbol for individual amino acid, okay? That means P stands for a particular amino acid, A stands for a particular amino acid, T stands for a particular amino acid, L stands for a particular amino acid, M stands for also a particular amino acid. That means you will have to, I am saying this, you will have to, memorize this single letter code, okay? It's really tough. What you are thinking is really tough, right? It's the most easy thing you will understand today. Listen. People don't discuss this because they are used to these things, okay? We will try to, put things in a very much, good manner. Listen. How many alphabets are there in English? There are 26, right? Out of 26, subtract 6, you will get 20 alphabets, right? Listen. There are 20 amino acids, no? That means each of the amino acid actually is represented by a single letter. Now listen, what are those letters? Listen, every alphabet is not representing an amino acid. What do you mean? Listen, there is A, there is no amino acid which is represented by the letter B. C, D, E, F. There is no amino acid which is represented by the letter J. Sorry. G, H, I, J. There is no amino acid which is represented by the single letter J. K, L, M, N, O. Right? There is no amino acid which is represented by the letter O. Then it is P, Q, R, S, T. There is no amino acid which is represented by the letter U. V, W. There is no amino acid represented by the letter X. Y. There is no amino acid represented by the letter Z. Okay? Now listen with all your attention. What is the full form of this A? This is alanine. This is alanine. What is C? Cysteine. What is D? Listen, this is aspartic acid. Now you are thinking that it is very tough. A for alanine, okay, no problem. C for cysteine, okay, no problem. But D for aspartic acid, how will you remember? Listen, it's a very tricky thing. Listen, aspartic acid, most of the case, you spell it as aspartic acid. Aspartic acid, right? That's why it is D. Okay. Now, glutamic acid. What? Glutamic acid is G for glutamic acid? No, G for glycine, but E for glutamic acid. How will you remember this? Very easy. Glutamic acid. Okay? Mic acid. Clear? Glutamic acid. F phenylalanine. Alanine. How will I remember? Very easy, FENYL alanine. G for glycine, everybody knows. H for histidine. I for isoleucine. K for lysine. What? Now L for leucine. Why are you doing this to me? Listen, Leucine was discovered first, that's why it took the letter L. Lysine came later, but the previous word, which was not associated with any, amino acid, was K. That's why lysine was given the K letter, single letter code. Okay. M, very easy. Methionine. N, asparagine. Okay. P, proline. You know what is the peculiarity of proline? Proline is an amino acid, not an amino acid, okay? It's an amino acid. And what is another important feature of proline? It's a helix breaker. Suppose there are proteins having alpha helices. If you introduce proline here, then it will break. That means proline is a helix breaker. Okay? Now listen. Q, glutamine. How to remember glutamine? Listen. Glutamine is equivalent to cutamine. Okay? Cutamine. R, very important, arginine. Why arginine was not given the name A? Because alanine was discovered first, and arginine R. If you remember this, arginine. S, serine. T, what? Yes, threonine. I was actually writing wrong. Threonine. V, valine. W, tryptophan. How will you remember tryptophan? You will remember it as tryptophan. Y, tyrosine, because T was taken and the next letter was Y. That's why tyrosine. Okay, clear. So these are the single letter codes you need to remember. Okay.